Okay, welcome everyone to virtual seminar. Good to see everybody here, and I'm very excited today to introduce today's speaker, Dr. Alex Payumo. A little background on Alex. He's been at San Jose State longer than his faculty position. He's an alum of SJSU who graduated in the class of 2008 after earning his BS in chemistry. He was a Mark Scholar and performed his undergraduate research with last week's seminar speaker, Dr. Darrell Eggers, where he studied the energetic contributions of water in protein folding equilibria. Motivated and inspired by his SJSU research mentors, Dr. Payumo then pursued a PhD in chemical and systems biology at Stanford with Dr. James Chen, where he applied novel optochemical probes to spatiotemporally dissect T-box transcription factor function during zebrafish embryogenesis. And I can't believe I got all of those syllables out successfully. He was an Iracta postdoctoral scholar with Dr. Guo Huang at the Cardiovascular Research Institute at UCSF. So he did all of his training locally, San Jose State, Stanford, and UCSF. And then when he was there, he was working on understanding the physiological mechanisms that limit the ability of the heart to regenerate in mammals. He joined SJSU's Department of Biological Sciences in fall of 2020. So his entire time as a faculty member has been inter-pandemic. But it sounds to me from talking to him like he's very excited to see people in person, just like I am, especially because of his commitment to training the next generation of scientists. Today, he's going to tell us about his work on the hormonal regulation of mammalian heart muscle cell proliferation and regeneration. And just a reminder to those of you in the audience, if you have a question, go ahead and type it in the chat. I'll keep an eye on it. If it's something that Alex should be interrupted for that needs clarification on the spot, I'll interrupt him. Otherwise, I'll let you ask your question at the end of the talk. So Alex, take it away.
All right, so thank you, Michael, and thank you for the opportunity to share with you a little bit about my research and what we're up to at San Jose State University. It's been virtual, it's been kind of crazy, but we've been trying to get stuff going, so it's my pleasure to share it with you and also pitch a couple things to you, like some technologies that we have that you may find useful and possibly open up ideas for collaboration. Okay, so I'm going to talk today about the hormonal regulation of mammalian heart muscle cell proliferation and regeneration. And this talk is going to occur in 3 pieces. First, I'm going to talk a little bit about thyroid hormones. Then I'm going to talk about thyroid hormone adrenergic signaling interactions. And then I'm going to transition to some of our current work that's ongoing at San Jose State University. So the first topic is on thyroid hormones. So heart disease is a leading cause of death in the United States. In fact, the CDC states that 1 in 4 deaths are attributed to this condition. So heart disease arises from a narrowing or blockage of blood vessels in the heart that occludes blood flow to heart tissues, right? So if you don't get blood flow to the heart tissues, that part of the heart dies. And this is called a heart attack. So this is bad. And the reason why it's bad is because adult mammals, like this mouse here, or humans like me and you, don't have an ability to regenerate their heart after injury. So if you took a section of this heart, like you cut a section of the heart and you stained it, the red stuff is all the cells that were living. And among those cells are these cells that beat, called cardiomyocytes. These are the heart and muscle cells. So that's why your heart's able to contract, act as a pump, and pump blood throughout your system. When you have a heart attack, a part of your heart dies. You know, you lose a lot of this red staining and it gets replaced by this purple staining. This purple staining is fibrosis and scarring. And now, since you've lost that staining, you've also lost a lot of those valuable cardiomyocytes, or those heart and muscle cells, permanently. So adult mammals lack the ability to replace these. And that's why it's such a critical condition if we were to lose them. Now, it turns out that while adult mammals typically are not very good at regenerating their heart, some vertebrates do possess an amazing ability to regenerate their heart. And this includes one of the upcoming kings of regeneration, such as the zebrafish. So as recently, well, not recently, about 20 years ago, Ken Poss discovered that if you were to take a pair of scissors and just cut a little bit of the apex off of this heart, over the course of about 2 months, that heart is able to completely regenerate without any fibrosis or scarring. So not like this. So this leads us to a paradigm such that perhaps heart regenerative potential is lost across evolution. And to illustrate this, I want to show you this diagram. So on the x-axis, you have different species like zebrafish, amphibians, rodents, large mammals, non-human primates, humans. And on the y-axis, you have cardiac regenerative capacity as a qualitative measurement, right? The height of this peak. So you can see while zebrafish and amphibians, or fish and amphibians generally have high abilities to regenerate their heart, this drops off considerably as we transition towards rodents, large mammals, non-human primates, and eventually humans. So that's an interesting question that we are potentially interested in trying to explore. So if we look at these animals that do possess the ability to regenerate their heart, such as the zebrafish, what lessons can they teach us about the mechanisms by which heart regeneration can actually occur? So if we took this zebrafish heart, so in this particular schematic, these rectangular cells, or those are those cardiomyocytes. These circular cells are like non-cardiomyocytes. They're cells that are in the heart that are not the cardiomyocytes. And then these two blocks here represent the apex. So using a genetic trick, so they add this drug called tamoxifen. They're able to label all the pre-existing cardiomyocytes and make them express GFP, turn them green. And what this looks like is like this. So all the cardiomyocytes in that zebrafish heart at this time, when they pulsed in tamoxifen, turn green. And if you're interested, you can look at the genetic construct, but it's not necessary to illustrate this point. So if you pre-label all those cardiomyocytes green, and then you take your pair of scissors and you cut this piece of the apex off, what these researchers did was ask, well, what color are the cells that form the regenerated tissue? And what I'm going to tell you is that those cells are actually green. So what does that mean? What that means to us is that the cells that form the regenerated tissue are actually derived from the pre-labeled cells, the pre-existing cardiomyocytes that were already in the heart. So this tells us something. So zebrafish teaches us that cardiomyocyte proliferation, the ability of those cardiomyocytes or those heart muscle cells, they need to be able to divide and make more of themselves for cardiac regeneration to occur. So this stresses the fact that regenerated cardiomyocytes are derived from pre-existing cardiomyocytes, and therefore we should start looking at how cardiomyocyte cell cycle control is regulated. Okay. So what if I told you is I kind of lied to you already? I said that mammals can't really regenerate their hearts, but they can sometimes. And recently, about 11 years ago, it was discovered, Enzo Perello and Eric Olson's lab discovered that if you take, so this is the same species now, this is a mouse, but if you take a baby mouse versus an adult mouse, and you take a pair of scissors, or you can do surgery and cause a heart attack, you'll kill part of the heart in this baby mouse. But over about a month, this heart is capable of 100% regeneration without any fibrosis or scarring. It's a very much different effect than, say, what happens in the adult. And these are the same, technically the same type of animal, same genetic code. The only thing that's different is the developmental stage, newborn to adult. So this suggests to us another paradigm, that maybe that cardiac regenerative capacity is also lost shortly or around the time of birth. So I'm going to draw another schematic here. So on the x-axis, you have developmental stages like the fetus. Here's at the time of birth. Here's 1 week after birth. And here's the adult. So you can see in rodents, if you look at this qualitative description of regenerative capacity, the ability of a rodent to regenerate its heart is high up until birth. And within 1 week, just 1 week after birth, the animal loses the ability to regenerate. And that's how things persist until adulthood. So that's also an important question. Why does that happen? So lessons from zebrafish tell us that the way that under normal conditions, the heart is able to regenerate is through the proliferation of existing cardiomyocytes. So therefore, what we should do is start focusing on what's going on at the cardiomyocyte level, those heart and muscle cells at these different stages through time. So when the neonatal mouse heart is able to regenerate its heart, those cardiomyocytes are mononucleated and diploid. That means they have one nuclei and they have two copies of the genome. So this is what cardiomyocytes in this heart look like during fetal and at birth. Within the first week of life, after the animal's born, 90% of the cardiomyocytes in this mouse heart transition to become binucleated. So they have two nuclei that are diploid, two nuclei that contain two copies of the genome. And the question is, why does that happen? To explain this to you, I want to remind you a little bit of the cell cycle. Like we've all heard of it, but it's useful to clarify, right? So remember, when the cell enters the cell cycle, enters G1, eventually it can duplicate or increase its DNA content during S phase. Then there's another resting phase that occurs after that. And then we get to mitosis. And there's a lot of cool things that are occurring during mitosis. So, the transition from interphase to prophase, you get condensation of the chromatin into chromosomes. Those chromosomes line up at the metaphase plate. Microtubules help to segregate like the chromatids. Okay, so during anaphase, now you get separation of the nuclei. And the last step in the cell cycle is cytokinesis, when the cell actually has to segregate its cytoplasm so it can form two cells. So why do we get this transition from a mononucleated diploid cell to a binucleated cell? Well, it turns out for cardiomyocytes, after birth, if those cardiomyocytes try to enter the cell cycle, they go through all of this stuff, but they arrest here. They can't complete cytokinesis. That's why we end up with a binucleated cell that has two diploid nuclei. So if we go back to our schematic from the previous slide, cardiomyocyte binucleation is caused by a failure in cytokinesis, right? Cytokinesis failure. This is lost across phylogeny or the evolution to mammals. Oh, my connection's unstable, maybe, hopefully, okay.
Yeah,
and
I think we lost the end of the previous slide. Could you?
This one?
Yeah, so you were talking about cytokinesis failure, and at least on my end, lost the very end of why that was the case. Yeah.
So the reason why, so the cardiomyocytes end up binucleated because they go through and enter the cell cycle, they go through mitosis, but they're not able to complete cytokinesis, that last step in mitosis when you're separating the cell into two cells. So they end up binucleated. They have the two nuclei, the nuclei separated, but not the actual cells. So this is the reason why we end up becoming binucleated. Okay, and there's two paradigms that we've brought up so far. First, heart regenerative capacity is lost across phylogeny or evolution. And secondly, is this age differential. Seems like in mammals that heart regenerative capacity may be lost across development, the development from newborn to being an adult. And why is this? And is there a common link? That was the motivating question when I started my postdoc at in Guo Huang's lab at UCSF. So what we noticed is that if you look at the animals we've talked about so far and we think about their cardiomyocytes and this transition from cardiomyocytes going from mononucleated diploid to binucleated, we see some patterns. So for example, in zebrafish, we can regenerate their heart. The majority of their cardiomyocytes, even as adults, remain mononucleated and diploid. Whereas in these newborn mice that can regenerate their heart, the majority of the cardiomyocytes are mononucleated diploid. But in this adult mouse that can't regenerate its heart, the percentage of mononucleated diploid cardiomyocytes is greatly reduced. So there seems to be a correlation with the mononucleated diploid cardiomyocyte abundance in the heart and the proliferative potential of the cardiomyocytes, which correlates again with the cardiac regenerative ability of these different animals. And this is validated by some, well, it's not so recent anymore, but by Michaela Patterson's work back in 2017, where she looked at a whole bunch of different mouse strains and examined what was the frequency or the percentage of these mononucleated diploid cardiomyocytes that occur naturally in a whole bunch of different mouse strains, over 100. So she's ranking the different mouse strains. So for example, these SWR and A strains have the higher percentage of those mononucleated cardiomyocytes. So after she identified these strains with the elevated levels of mononucleated cardiomyocytes, they did surgery on them. Those strains tended to recover better after injury. So this gave us an idea that perhaps you can think of the percentage of mononucleated diploid cardiomyocytes in the heart as a proxy to gauge whether or not an animal could regenerate or not. So as a postdoc, what I did was we extended this idea, but instead of looking at many strains of mice, we went a little broader. We were looking at mononucleated diploid cardiomyocyte abundance as a proxy for heart regenerative capacity, but across different animals now. So we have our standards. We know what's going on with zebrafish. We know what's going on with mouse. So what we can do is we can take a piece of their hearts, dissociate those cardiomyocytes into single cells, sustain it with a nuclear dye like DAPI. That allows you to see how many nuclei are in each of those cells. So here in the white arrows are all the mononucleated cells. So what if we did that, but we took it from a lot of different, we sampled a whole bunch of different animals, ranging from lizards, bats, echidna, platypus, armadillo, a sloth, a couple whales, et cetera, et cetera. Just sample across different animals. What do we get? What kind of patterns can we see? So when we did that, at the end of the day, we analyzed cardiomyocyte nucleation, the percentage of diploid cardiomyocytes across 41 different animals, 6 non-mammals and 35 mammals. So if you're interested, for example, in the mouse, this is what the cardiomyocytes look like. They're more blocky, right? Versus in the platypus, they tend to be more elongated. And that's actually an interesting trend. So don't ever think that because an animal has a heart that all the cells look the same. Cardiomyocytes may look very different, you know, at this end of the spectrum versus this. And then I was also interested in the size of the bowhead whale and sperm whale cardiomyocytes. I just thought maybe the cells would be super big, but they're not. They're actually similar in size. I guess they just have more of it. So this brings up some other interesting questions on how like cell size is regulated. The morphology of cells with respect to their function is also interesting. Okay, but at the end of the day, what we found, for example, in the mouse is we validated what's already known in the literature that adult mice tend to have about 10% of these mononucleated diploid cardiomyocytes. But we found a whole bunch of different, what you would consider to be mammals that have pretty much had a higher elevation of these cells, 40 to 65%. So like the bat, bowhead whale, platypus, armadillo. So it begs the question. So if you're thinking about the abundance of diploid cardiomyocytes in the heart as a proxy to gauge cardiac regenerative capacity, hey, these animals that you find that have elevated amounts of cardiomyocytes, do they have more proliferative potential? Do they have more cardiac regeneration? So that opens up the idea is maybe we should be more broad in trying to investigate heart regenerative capacity across different types of animals as well, other than more traditionally model organisms, right? Like zebrafish or mouse. There may be secrets in these transitional species, if you would. So what we're interested in finding also is that we had all this data, but are there any physiological traits that correlated with this pattern that we saw? So we tested things. So for example, we said, okay, how about like just the average body weight of an animal? Does that seem to track with their mononucleated diploid cardiomyocyte frequency? No. How about their natural heart rate? Maybe that's a proxy for how hard the heart has to work. No. How about the pressures in the heart when it's contracting, systolic pressure? No. Diastolic pressure? No. So we tested a whole bunch of things, but we didn't see a lot of correlation between traditionally like physiological factors and this correlation with diploid cardiomyocyte abundance. But after some searching, what we ended up seeing is a very strong correlation between diploid cardiomyocyte abundance and standard metabolism, standard metabolic rate. And what we found is an inverse correlation. So on this graph here, you have on the x-axis a measurement of standard metabolic rate. And on the y-axis, you have the percentage of diploid cardiomyocytes. And if you can see from this pattern, animals that tend to have lower metabolism tend to have higher percentage of these diploid proliferative cardiomyocytes. And on the other end of the spectrum, animals that have higher metabolism tend to have lower amounts of these diploid cardiomyocytes. The other thing I want to point your attention to is a lot of the animals on this side of the graph are ectotherms or cold-blooded animals. They don't have the ability to self-regulate their body temperatures. And then as we're moving along this line, we start getting to endotherms or warm-blooded animals, the animals that can maintain constant body temperature. So there may be some implications in an ectotherm to endotherm transition as well, if evolutionarily.
Hey, Alex,
Yeah.
can I interrupt with a question?
Sure.
So where would the zebrafish be on this plot?
It would be up here somewhere.
Way up there. Okay, so the metabolic rate is some kind of let's see, is that watts per kilogram? Is that the measurement?
This is standard metabolic. So this is modeled from Kleber's law. I think it's basal metabolic rate is equal to standard metabolic rate times mass to the 3/4 power.
Okay, so so this is normalized to mass somehow.
This is normalized to mass.
Yeah. Okay.
To the 3/4 power though, which is really interesting too. So if you're interested, there's a law that says this relationship, and it's able to predict animal metabolism across, multicellular animals like elephants, huge elephants,
right,
to unicellular animals, down to individual respiratory complexes too.
Right. Yeah, I've seen those plots before. I just never really paid attention to the units of this. So cool. Thank you.
Yeah. Okay, so we're looking at this inverse relationship between metabolism and the frequency of these diploid cardiomyocytes. And we're thinking, okay, there seems to be a pretty strong relationship between metabolism and this proxy for heart regenerative capacity. So we're thinking then is like, what molecular pathways could be driving this? And one of the things that came to our mind, I mean, it's a longer story than this, but for now, we'll just say it's one of the things that came to mind, are thyroid hormones, because they're known as classical regulators of metabolism. So thyroid hormones are secreted from the thyroid gland, and there's two major forms. There's T3, which is the more potent form that can bind the thyroid hormone receptor, and also T4, which is more abundant, but it's more considered like the prohormone that needs to be processed into T3. Traditionally, this signaling or this endocrine signaling is important for increased metabolism, as well as for stimulating growth and development. And interestingly, classical experiments using thyroidectomy, so they actually cut out the thyroid gland in these different animals, show a dependence of animal metabolism on thyroid function. For example, if you take a rabbit as a representative of the eutherians and you perform thyroidectomy in those experiments, the standard metabolism of the animal is going to drop. But if you take this bandicoot, which is representative of marsupials, standard metabolism is going to drop. But interestingly, for this monotreme, so this echidna or the egg-laying mammals, their metabolism doesn't seem to be dependent on thyroid hormone function. So thyroid hormone function and its stimulation or support for metabolism may be an innovation in, these animals. So somewhere there seems to be a divergence in how thyroid hormone supports metabolism. Maybe it's a newer innovation. So the other thing is, okay, maybe there's this link with evolution, but how about during this other paradigm of development? So what we've done also, looking at newborn mice to adult mice, we measured thyroid hormones, for example, T4 and T3 at birth and then over the first week, and there seems to be an increase there. So it seems to be shared across animal development as well. So how can we test this then? How can we formally test this idea? that maybe these increasing levels of thyroids are causing the cardiomyocytes to not be able to proliferate and binucleate and arrest in the cell cycle. Well, we got to talk about a little bit about how thyroid hormones work. So thyroid hormone receptors are traditionally nuclear receptors. They already live in the nuclei and they bind to these TRE elements. And without that presence of a ligand or something that binds to that thyroid hormone receptor, it usually recruits co-repressors. So without ligand, thyroid hormone receptors are actually keeping their genes suppressed, they're inactivated. And once you have thyroid hormone bind to a thyroid hormone receptor, they can remove or release the co-repressor and recruit co-activators to turn on their gene expression. Okay, so how are we going to test this? Is we're going to take advantage of a system that mouse biologists use a lot, the Cre-Lox system. And just to review, Cre is a recombinase. So if you use genetic tricks or sequences like the LOXP sites, in the presence or the directionality of the LOXP, you can get recombination. So you can cut a piece of DNA out or flip its orientation. But you can use this technique, for example, if you had a tissue-specific promoter that would drive expression of the Cre in only a cell type that you're interested in. So for us, for example, cardiomyocytes. And you breed this line with something else that you wanted to either express a gene or delete something else. There's like different genetic tricks that you can use. You can allow tissue-specific expression of a gene of interest. So I'm going to explain it, but not too deeply. So we use a mouse genetics approach where we take a MyH6 Cre. So we're using a cardiomyocyte promoter to drive the expression of Cre. And we crossbreed that to a line that in the presence of Cre, you're going to overexpress a dominant negative thyroid hormone receptor. So these thyroid hormone receptors will be able to bind to those TRE elements, but cannot recruit activators. So they'll outcompete the endogenous receptors and therefore make sure that those genes that would have get turned on in response to thyroid hormone cannot get turned on. So this is going to be our genetic trick. We're going to overexpress a dominant negative thyroid hormone receptor specifically in the cardiomyocytes, in the animal. So we'll be able to study tissue-specific control in the animal itself. So when we create these mutants, so here's the one where we were messing up thyroid hormone signaling in the cardiomyocytes. Here's your controls. One of the things that we notice is that the heart is actually a lot bigger. And there's a lot of reasons why the heart could be bigger. It could be there's more cardiomyocytes, but it also could be that the heart undergoes hypertrophic growth, which means that the cells just get bigger, but you're not actually making more cells, right? But one of the things that happens in these mutants, the heart gets bigger. So here's the body weight stays the same, but the heart weights are bigger. So therefore the heart weight to body weight ratio is bigger. But if you look at the cardiomyocyte level, you'll actually see if you quantify the number of cardiomyocytes in the heart that there's more of them. Not only that, but the percentage of those diploid mononucleated cardiomyocytes that we think might be proxies for increased regenerative potential, those are increased. So you're around 10% in the controls. Now you've increased that to 30% in these mutants. And if you measure the volume of the cells, they seem to be decreased. So it's not hypertrophy that's leading to the increased size of the heart. Now, if we take the hearts out of these animals and section the tissue, we can actually try to stain for cardiomyocyte nuclei. So that's what PCM1 is. Those are all the cardiomyocyte nuclei. And also cell cycle markers that we traditionally know, like Ki67, which is expressed in all cells which was expressed in all stages of the cell cycle. So when you see co-localization of these green dots with these red dots, that means these are cardiomyocyte nuclei in the cell cycle. And what we find is in these mutants, there's more of those cardiomyocyte nuclei in the cell cycle. So they seem to be in the cell cycle more. So all of this together suggests, wait, I think I missed some. Oh, no, I told, yeah, now we're good. So we see more cell cycle activity. We see more diploid cardiomyocytes. So obviously what we want to test is if these animals can regenerate. So to do that, we do a surgical model called ischemia reperfusion injury. And how this works is we take an adult mouse and you have its heart, and there's a major vessel in the heart called the left anterior descending coronary artery. And to do an ischemia reperfusion injury, you basically tie a suture around this major branch. And then ischemia reperfusion means you only do it temporarily. So you tighten the suture just for a little bit, cause ischemia or a lack of blood flow to the tissue. then you release it. And we usually do that after 45 minutes. So that's the reperfusion part. Now the blood can go back into the tissue, and that also causes damage as well. So we're using a ischemia reperfusion injury in this example. And what happens when you do that, so 10 days after the injury, if you take sections again, what we see next to the injury site, so that's this dark area that's beyond the dashed lines, increased again cardiomyocyte cell cycle activity, so more green and red dots that co-localize close to the zone of that ischemia reperfusion injury. So there seems to be more cell cycle activity after injury. In addition, if you take sections of the heart and stain for fibrosis or that scar tissue, we see in these thyroid hormone mutants, so these are the cardiomyocytes that can't respond to thyroid hormone signals, there's less fibrotic area or less scar tissue. And finally, what we can do is use an ultrasound technique or an ultrasound-based technique called echocardiography to measure how the heart function is working in the animals based on the distance of the walls when it's contracting and relaxing. So you can use echocardiography to measure ejection fraction, which is a measurement of heart function. And what we see is in the animals that are control, these are the black animals, after you do the ischemia reperfusion injury, heart function will decrease and pretty much stays decreased over the course of 28 days. Whereas in the animals where you've inhibited thyroid hormone function, it will decrease, but eventually it can start rising up again. So this is in response to ischemia reperfusion injury. So for these, and what these experiments tell us is these evidence are pointing to a model. And I'm going to explain what this model could be, suggesting that across evolution, perhaps there's increasing levels of thyroid hormones that are in the circulation in these animals in order to drive metabolism, right? Especially maybe this idea of being cold-blooded to warm-blooded. However, these increasing levels of thyroid hormone may be doing something at the level of cardiomyocytes. So low levels of thyroid hormone may be permissive to allow cardiomyocytes to enter the cell cycle and divide. Whereas higher levels may be promoting cell cycle arrest or binucleation. And this may be the reason why, an adult mouse no longer can regenerate its heart versus a zebrafish can still regenerate its heart. So the model is elevation in thyroid hormone levels increases energy metabolism in these animals, but it may decrease cardiac regenerative potential as a trade-off. So there's an inverse relationship with the ability to regenerate the heart, but perhaps drive your metabolism to support to drive metabolism in mammals. And the question is why? So I'm going to leave this with a quote from Richard Goss, who's a regenerative biologist in 1969. So what he says is, it seems that regeneration is not necessarily adaptive, but that it has disappeared or persisted as other more important physiological attributes evolved, with which it may or may not have been incompatible. So it's not necessarily that regeneration is a necessary thing. It's just maybe we need more traits that outweigh the importance of regeneration. And so, okay. So hence, before any intelligent attempt can be made to restore regeneration where it does not normally take place, it will first be necessary to learn what physiological advantages took precedence over regeneration in the course of natural selection. So again, maybe the ability to regenerate the heart is not a necessary thing. I mean, we still are able to live our lives without the ability to regenerate our heart. But how about humans? Are we going to be able to live our lives if we can't regulate our body temperature? Are we going to be able to colonize different environmental conditions, cold, hot, high altitude, without being able to maintain homeostasis, that constancy of body temperature? So again, maybe this is suggesting to us that as these physiological attributes like endothermy or warm-bloodedness evolve, those things are not molecularly, those things are not compatible with the ability to regenerate our heart. So it's a trade-off. Okay, so that's the idea I want to leave you with there. So we're starting to get to the next part of the talk, and I won't, the other part will not take as long. But it's likely that other pathways will work with thyroid hormones, because if you look at the data that I showed, if you caught it, when you inhibit thyroid hormone signaling in these animals, 30% of the cardiomyocytes are diploid cardiomyocytes. Only 30, right? That means 70% of the cardiomyocytes still became binucleated. So it's unlikely that thyroid hormone is the only pathway. So this leads us to the next part, thyroid hormone and adrenergic signaling interaction. So this is work that we've recently tried to wrap up for my postdoc as I was transitioning here to San Jose State. So there's a lot of physiological changes that occur after birth. Some of it is plasma hormones like thyroid hormone, they increase after birth. But another thing that we're interested in focusing on is sympathetic nerve activity. So this is the nerves that, there's the sympathetic nerves that are activated in your fight or flight response, right? So that's why your heart's beating really quick. That also starts getting activated after birth, okay? So sympathetic nerves actually innervate the heart before birth. So you can see based on tyrosine hydroxylase staining that sympathetic nerves are already present by E14.5. So this is embryonic stage 14.5, okay? However, the connectivity or this circuit of sympathetic activity seems to be active after birth. So for example, for sympathetic nerves, you have your preganglionic neurons that interface with postganglionic sympathetic nerves that innervate the heart. So at this synapse, for example, you can test this connectivity by stimulating with nicotine and looking for expression of downstream target genes in the downstream tissues, like ornithine decarboxylase. So this is an enzyme. So an old experiment that they did is they stimulated nicotine at different points in development to see if ornithine decarboxylase activity would increase in the downstream tissues, to see if this sympathetic circuit is active. And then they assay for the ornithine decarboxylase activity, and they only see net positive gains in activity after birth. So it seems like the nerves are there before birth, but maybe they're only really active after birth. This is from rat. So sympathetic nerves activate adrenergic receptors. There's a lot of adrenergic receptors. So there's alpha adrenergic receptors. Well, there's two major families. There's alpha adrenergic receptors, and there's 6 subtypes. So it's kind of complicated. We don't need to get into it too much. And there's also beta adrenergic receptors, and there's 3 subtypes. So the most relevant alpha adrenergic receptor subtypes are probably alpha 1A and alpha 1B. And then the most relevant beta adrenergic receptors in the cardiomyocytes are beta 1. People used to think beta 2, but there was the recent paper that suggests that cardiomyocytes actually don't express beta 2. So there's a subset of all of these different adrenergic receptors, but we want to focus in ideally on the ones that are relevant in cardiomyocytes. So sympathetic nerves release neurotransmitters like norepinephrine that activate both alpha and beta adrenergic receptors. So what we wanted to see is if, so in the first part of the talk, we discussed thyroid hormones. We think that they're important for driving cardiomyocytes out of the cell cycle, as a trade-off, again, to stimulate endothermy. Now we wanted to test whether or not there's interactions between thyroid hormone signaling and adrenergic receptor signaling. So because the receptors are so complicated, it's hard to narrow it down to like one receptor. So we decided to go with a pharmacological approach. So let's say we have our transition from newborn to 2 weeks old. We have a bunch of pharmacological inhibitors for adrenergic receptor, as well as thyroid hormone signaling. So alpha adrenergic receptor inhibitor, we were using phenoxybenzamine. Beta adrenergic receptor inhibitor, we were using propranolol, which is an old school beta blocker. And the thyroid hormone inhibitor, we were feeding the animals with propylthiourea, which inhibits production of thyroid hormone. So we treat these animals with all of this, so basically a cocktail of these drugs in combination, and asked, well, what is the effect on cardiomyocyte cell cycle? So for shorthand or clarity, these are the codes that I'm going to use. So black, green, blue, and red. Black is control. Green is going to be adrenergic receptor, so both alpha and beta together. PTU is just going to be thyroid hormone inhibitor alone. And then red is everything together. So consistent with our idea in terms of body temperature, what we found is when you treat animals with the triple cocktail. So I forgot to mention, so like during the first 2 weeks after birth, so baby mice are not born warm blood. They're poikilothermic. They require their mother to maintain body temperature. And over the course of 2 weeks, under normal conditions, they will be able to self-regulate their body temperature, but it takes about 2 weeks. So if you treat these animals and then examine their body temperatures with a thermal camera, if you look at the triple combination, you see a large suppression in ability to maintain their body temperature versus thyroid hormone alone or adrenergic receptor signaling. So that's something that we noticed, that there was definitely synergy in terms of inhibiting the animal from maintaining its body temperature. If we looked into the cardiac tissue again, using the same assays from before, so all the green dots are cardiomyocyte nuclei, all the red dots are cardiomyocytes in the cell cycle. What we see is this versus like thyroid hormone inhibition alone, the combined effects has some more, like there's additive effects. They seem to be working together. So here's adrenergic receptor signaling alone, there's thyroid hormone signaling alone. Together, you get a much bigger bump in the percentage of cardiomyocytes in the cell cycle. How about the diploid cardiomyocyte content? So if we use the same assays as before, we isolate the cardiomyocyte stain for the nuclei, try to calculate the frequency of those binucleated diploid cardiomyocytes. Again, the combination of those pathway inhibition alone, or together, is far greater than either adrenergic receptor signaling inhibition or thyroid hormone inhibition. And finally, the next question would then be, how about their regenerative capacity? Okay, so this is a little bit of a different model. So before I was talking about ischemia reperfusion, where you temporarily tie off the left anterior descending coronary artery. Now we're going to do a full MI. So that means you just completely tie off the vessel together. So I'll tell you that those thyroid hormone mutants that I talked about earlier, they do not regenerate after a complete MI. So this is a more severe injury. So now we're testing that in these mice. So we're going to do the complete MI and then treat with the drugs from P0 to the end of the assay. And what we see in this particular case is only the animals that were treated with a cocktail of thyroid hormone and alpha and beta adrenergic receptor inhibitors were able to recover after complete myocardial infarction. Okay, consistent with that is the percent of scarring and fibrosis. So now here's sections of the heart. Here's the apex of the heart, and this is where the suture was tied. You can see there's a lot of dead tissue in the controls. It's a lot bigger than ischemia reperfusion because again, MI is huge. And here's the alpha adrenergic and beta adrenergic inhibitors. Here's the PTU, thyroid hormone by itself. But you see there's a lot less scarring tissue in the cocktail. So it seems like thyroid hormone definitely has an improved effect on its own, consistent with this idea that maybe thyroid hormones are driving metabolism. When you combine thyroid hormone plus adrenergic receptor inhibition, that even suppresses the ability of animals to maintain their body temperature further, and it seems to enhance further their ability to regenerate their heart. Now, the current challenge that we're facing, or I'm facing, now that I'm transitioning to more of an independent position at San Jose State, is we don't know the downstream mechanisms for any of these pathways. And I think part of that is all the work that we've talked about so far had been in an animal. And it just turns out that animal physiology is pretty complicated, right? Here's my animal. The previous experiments that we just did, we were injecting all these drugs into the animal. There's a lot of organs in there, but really we're focusing on just the heart. So if you have this systemic injection of drugs throughout the animal, and other tissues express adrenergic receptors, other tissues express thyroid hormone receptors, what's the mechanism? In fact, I don't even know at this point if these signaling pathways are working directly on the cardiomyocytes themselves or through some other indirect physiological signaling. I don't know at this point. So that brings us to our last part of the talk, which is not going to take super long. But what is the current work that our lab is trying to do at SGSU? So to clarify some of these mechanistic unknowns, we're going to take advantage of a much simpler primary rat cardiomyocyte culture model. So instead of working with the animals in totality, we will take newborn mice or rats, and we're going to isolate their hearts. and dissociate the cardiomyocytes out of the hearts and then culture them in a dish. That simplifies the system. No more animal, just the cardiomyocytes in a dish. This allows us then to treat directly with drugs that activate these pathways, alpha or beta adrenergic receptor signaling or thyroid hormone, and assess what is the direct effects of these hormone activation of these pathways on cardiomyocyte behavior. So we'll take the animal out of the equation, we'll be able to see if there are direct effects. So what our lab has been up to over the past year and a half or so is we figured out how to culture the primary cardiomyocytes. So these are done at San Jose State. So for example, if you've never seen cardiomyocytes, this is what they look like. So we're using serum-free culture. You can see if you don't add anything to the cardiomyocytes, they don't beat so strongly in our serum-free conditions. But once you add or activate thyroid hormone adrenergic receptor, they start beating like crazy. So that's always cool. We've also optimized a serum-free cardiomyocyte proliferation assay. So we use antibodies to detect cardiomyocytes in our dish. We treat with, or we can detect cell cycle markers like incorporation of EDU, which labels the cells in S phase. And we can see that, oh, and then we also, because it's serum-free conditions, the cardiomyocytes don't really want to enter the cell cycle and divide without serum. So if you guys are not familiar with cell culture, serum it provides cells that you're growing with growth factors or things that allow them to proliferate. But in our particular case, we're not using serum because we're testing out the function of these hormones, and these hormones are present in serum to some unknown degree. So it would complicate our situation. So we've and this is with the help of Dr. Gorilla Hill's lab, who does a lot of Wnt beta-catenin signaling, but we are using a chemical inhibitor of GSK3, which we think is activating Wnt beta-catenin signaling to stimulate the cardiomyocytes to enter to the cell cycle. So we made a serum-free, chemically defined way to push cardiomyocytes into the cell cycle. And when we do this, we see evidence of mononucleated and binucleated cardiomyocytes in culture, which is good. Our preliminary results so far suggest that if you do treat these cardiomyocytes with activators of adrenergic receptor signaling or thyroid hormone signaling, or in combination, that it will inhibit those cardiomyocytes from dividing. So it seems to recapitulate at least that part in vivo. So I think we're okay with this primary cardiomyocyte model. I do want to introduce this because this could be a point of collaboration if people are interested, but our lab uniquely has, this is a new technology. It's called digital holographic phase imaging. This microscope is really small and lives inside the incubator. And basically the way it works is it has a sample and a reference beam. So the sample beam passes through your sample and the reference beam is used as a point of reference. And then the interference pattern between these two beams creates this pattern, which then the software can calculate and reconstruct a three-dimensional image of what that cell could look like. So this is called digital holographic phase imaging. We are trying to pioneer its application to study cardiomyocyte behaviors right now. And so far, for example, we can culture our rat cardiomyocytes and then image them using this technique. And we can see evidence, for example, here's a cardiomyocyte that's about to divide. We can see that. We can also see evidence where they binucleate. So we'll be able to see the dynamics of perhaps I figure out some patterns or something, or I don't know. We need to be able to see, because it's very hard to get this level of resolution when you're looking inside an animal, right? So by culturing the cells, applying this technique, we may be able to get further insights on what's going on, okay? And the next part, so this is from a cellular angle. We are also very interested in the molecular downstream targets of these hormones. We don't know that quite yet. So we've also at San Jose State started doing some RNA-seq experiments where we compare control cells versus cells that we've activated thyroid hormone and adrenergic receptor signaling. Consistent with our observations, we do see a lot of cell cycle related genes that are downregulated. So these are genes that are important for nuclear division, mitotic nuclear division, chromosome segregation, et cetera, et cetera. So it does seem like the ability for these genes to enter the cell cycle is suppressed. So it does give us an opportunity to start exploring, like, what are the cell cycle inhibitors downstream of these hormonal pathways that are suppressing cell cycle from a molecular standpoint. So essentially what we're trying to do at San Jose State is use the momentum that we have from the in vivo experiments, but now start looking at the cellular and molecular basis of what these hormones are doing in terms of controlling cardiomyocyte cell cycle. So essentially, if we force cardiomyocytes to enter the cell cycle, like what is their logic? Like why do some divide? Why do some binucleate? There's even a third option that I didn't really talk about, but it's called endomitosis, where instead of separating their nuclei, they duplicate their DNA, but they have one big nuclei. And actually, human cardiomyocytes don't look mostly binucleated. They look like this. So there's this trend for increased DNA content, but in humans, most of the cardiomyocytes look like this. So what's the reasoning? What's the logic between why when a cardiomyocyte enters its cell cycle, it resolves in these different ways? And how do these hormones affect that resolution? Those are some of the questions our lab is currently interested in. So acknowledgements, I'd like to just thank the Huang Lab at UCSF. So this is Gua Huang, looks very young, very kind. He's actually really great. Now he's been very supportive of me, even he now at San Jose State. I still talk to him from time to time. And then here's Kentaro and Shi, who were the co-first authors in the thyroid hormone and also the thyroid hormone adrenergic paper that we talked about. And this is my current lab at SJSU. So right now I have two awesome graduate students. So Kurt and Ines, somewhere here, and then a bunch of undergrads as well. So our lab is pretty big now. So we have two graduate students and I think maybe 11 or so undergrads, and they've been great. So I've been enjoying myself working with them, and like they're learning how to do these cardiomyocyte cultures, some molecular cloning now, imaging. So it's been fun. And I'd finally like to just thank C-Superb. So how are we getting funded at the moment? Well, C-Superb New Investigator Award has been helping us out a bit, and also a lot of help from the SJSU RISCA seed grants, and also a grant that I had written with Nanak. That's also helping us out a bit. So that's all I have. So I'm sorry a little bit over. I wanted to leave 10 minutes, but we have about 5 minutes, but I'm more than happy to answer any questions or clarify anything at this point. Thank you.
Thanks, Alex. Fascinating. I'm a novice in this stuff, but I think you started to hint at it a little bit. I assume that one of the potential applications is regeneration. You talked about it.
Right.
And I'm curious about what the state of the art is in modifying existing cells in order to allow for regeneration to possibly happen.
I think there's a lot of approaches in regeneration right now, right? So the approach that we're taking is try to understand what are the locks to natural regeneration. Right. There might be clues in that developmental transition, right? From neonate to adult. So if you figure out what's locking down the ability of the cardiomyocytes to divide, you might be able to unlock that ability in an adult. That's the hope. There's other strategies too. You could also do transplants. So people have been trying to reconstruct the heart, or people are doing like stem cell based approaches too.
Right.
So there's a lot of different ways to skin the cat. There's also bioengineering ways too, like you can make a sleeve that covers the heart and allow the heart to rest. And then, from an engineering point of view, mimic the contractility.
Right.
So there's, yeah.
It's amazing work. And the other thing is, you alluded to this, that there isn't necessarily an evolutionary advantage to either keeping or losing the cardiomyocytes. But I just wonder if you would speculate about what, suppose it was an advantage one way or the other. Can you think of any good reason why the higher metabolism animals lose the cardiomyocytes?
Well, I mean, the higher metabolism animals, there needs to be a reason of why they need that metabolism. And what we think right now is because you're warm-blooded, you need a lot of energy, even just to, like, through brown fat mediated thermogenesis, just to get enough heat production so you can maintain your body temperature. So that's very different than if you're a cold-blooded animal that needs to bask in the sun or something to regulate your body temperature. Your body doesn't have to do as much.
Right.
But that's an important thing for us, or else we'd have to bask in the sun.
Mhm.
And we wouldn't be able to live in cold environments or
Right.
dark environments. So from a survival advantage, the ability to be warm-blooded may outweigh any kind of need to regenerate the heart.
Got it.
Right.
I also wonder, I mean, pre industrial revolution, how much death in humans was because of heart failure, and how much was because of things that,
well, people are also living a lot longer now.
Yeah, well, that's kind of the point I'm making, is that maybe heart regeneration wasn't such an important advantage.
Yeah, when the average lifespan is like 40.
Right.
Yeah, it could be.
Anyway,
I think all of those things factor in. Yeah.
Okay, I'm going to hand it over to some other people. Let's see. So there's an anonymous attendee, which I think means I can't turn on their sound. So I will just go ahead and ask the question. If an animal has a malfunctioning thyroid gland, would regeneration still be possible?
I think that's a good question. And it's something that we haven't tested. But yeah, in disease states, or what we call them disease states, right? If you're hypothyroid, do you have a natural ability or an enhanced ability to regenerate their heart? I don't know. We haven't tested it yet. But also, you got to think about how permanent is that developmental transition. So when you're newborn to an adult and you had high levels of thyroid hormone and that cell cycle activity shut down, is it reversible? So maybe if you were born thyroid depleted, maybe you would have enhanced ability. But could we, for example, acutely just drop thyroid hormone levels? And would that facilitate heart regeneration in normal patients? I don't know. It depends on how plastic or how set in stone the development is, and how reversible things are.
We have one question from Myra Cardenas. Myra, you should be able to talk, unless you just dropped off the call. I'm not sure. Yeah, no, she's there. Go ahead and unmute yourself, Myra.
Hi. Wondering if you're taking any more students to join a research lab?
I have a bunch of students that may be graduating this semester, so I might be taking a few for fall. But I think, yeah, send me an email. Or if you go, we have a lab website with a contact Google form. And if you're interested, just fill it out and I'll get back to you and we can arrange a time to chat.
Thank you.
All right. I don't see any other questions in here, and we are at the hour. But if, Alex, are you able to join us for a little happy hour afterwards?
Yeah, I can go. I saw one question. Can the levels of...
Oh yeah, there's one more question here. Also from the anonymous attendee, can the levels of thyroid hormone from infancy be regulated as the animal grows?
I mean, theoretically you could. There are drugs that allow you to regulate thyroid hormone levels, but you got to keep in mind that thyroid hormone is important for a lot of different other tissues. So if you noticed in the animals that I showed you, some of those pictures that heart looks small, that's because the animal was small. So like from a developmental standpoint, a lot of tissues require the thyroid hormone. So if there was a way to, possibly prevent the cardiomyocytes specifically responding from the thyroid hormone, that might be safer. Other than that, you might be affecting other things that you need to develop as well.
Great. Okay, well, thank you, Alex.
Yep.
Great talk. So cool to see your lab up and running.
No, it's been fun. Yeah, no, it's great.
growing creepy heart muscle in dishes.
It's not creepy.
No, it's awesome. It's really cool.
Okay.
I would love to come and see it sometime, actually.
Yeah, absolutely. So is this the link or?
No, I emailed it out right after the talk started, so you should see it in your email. And thanks everybody for joining us. Thanks again, Alex. And if you're interested in joining Happy Hour, there's a link in the email that you should be able to follow. See ya on the other end.
See ya. Thank you.